Dear audience, it's a great honor to be here today and to give a presentation. Thank you for the opportunity. In the following couple of minutes, I would like to present my research journey over the past 2-3 years. So I'm going to talk about an optimal ischemic retinopathy mouse model and the consequences of oxygen deficiency in the retina. But how exactly a hypothesis is formulated, and how can we get from the question to conclusion? After a thorough literature review and based on previous experiments, we find an unanswered question. Then we plan the methods and get the results. After interpreting the data, conclusions are drawn. This figure shows the idealistic path, but let me show you the real one. At this point, you may wonder why we choose the previously mentioned topic. The retina has one of the highest oxygen consumption and metabolic activity, so lack of oxygen supply easily leads to visual impairment. Diabetic retinopathy, age-related macular degeneration, glaucoma, and retinopathy of prematurity. These diseases have very different etiologies, however, they are all caused by the hypoxic state of the retina. Despite the prevalence and the severity of these conditions, we have no effective side effect-free treatment options. In addition, the pathomechanism is not fully understood. That's why our research group aimed to better understand these diseases and to find new potential therapeutic targets. But before I get into the details, let me show you an overview of the retina structures. And I would like to highlight the ganglion cell layer and the photoreceptor layer. The cell bodies of ganglion cells are found in the ganglion cell layer, while the inner and outer segments of rods and cones are in the photoreceptor layer. Now let's see the blood supply of the retina. The inner 2/3 are supplied by the central retinal artery, which comes from the ophthalmic artery, which is a branch of the internal carotid artery. And the internal carotid artery originates from the common carotid artery, while the outer third contains no blood vessels, and it is supplied by the choroidal vessels by diffusion. And the choroidal artery is also coming from the common carotid artery. Based on the literature, the occlusion of the mentioned common carotid artery for 10 minutes leads to a mild retinal ischemia. So that's why we plan to use this model for our further investigations. So after this procedure, you may wonder what happened. Almost nothing. So that's why we have to dig deeper into the literature to find a new model. But the available ones were not appropriate for our aims. That's why our aim was to develop an optimal ischemic retinopathy mouse model to investigate the retinal damage in a time-dependent manner. Now let me show you our methods. Retinal ischemia was induced by bilateral or unilateral common carotid artery occlusions. So BCCAO was performed for 10, 13, 15, and 20 minutes. Furthermore, permanent unilateral common carotid artery occlusion was performed, and sham-operated animals served as controls. After the surgeries, an optical coherence tomography measurement were performed. So OCT is an in vivo pill-less method which is widely used in a clinical practice. And after taking the snapshots, the software analyzed the layers and measured their thickness with auto-segmentation. So these pictures show our experimental setup. And OCT measurements were performed before the surgeries and 3, 7, 14, 21, and 28 days later. So in conclusion, our analysis included more than 5,000 data from around 400 measurements. So after taking the snapshots, a complex standardization method was performed, and the linear random effect mixed model was used as statistical analysis. So this is just for validating our method, and you can see that the individual values distribute randomly around the horizontal line, which means that we can use our model for further analysis. So 28 days later, all retinas were isolated, and immunohistochemistry was performed together with other molecular methods on whole mount retinas and on cross sections. You can see the steps of our procedure in these pictures. Now let me show you our results. Begin with the OCT ones. So all retinal layers were analyzed separately, but I would like to highlight just the total retinal thickness because of the limited time. So you can see all groups separately, and the horizontal axis represents the days after the surgery, while the vertical axis shows the standardized thickness results. And we experienced a significant decrease in the total retinal thickness after any kind of ischemia, but the damage was the most severe in the UCCAO group. GFAP, or glial fibrillary acidic protein, is a commonly used general retinal stress marker. GFAP labeling was performed on whole mount retinas and on cross-sections, and you can see all nuclei with DAPI staining. Our immunohistochemistry results were quantified by Western blot analysis, where we experienced a significant increase in the GFAP expression after UCCAO compared to controls. Furthermore, all retinal ganglion cells were labeled and analyzed in the central and peripheral regions of the retina. So the upper picture shows the central, the lower picture shows the peripheral regions, and the red dots are the individual ganglion cells. So you can observe the decrease in the number of the ganglion cells after 20 minutes BCCO and in the UCCO compared to controls with the naked eye. And after quantification, we observed a significant decrease in the ganglion cell number after 15 and 20 minutes BCCO and in the UCCAO group in the peripheral regions of the retina. The tendency was similar in the central region, but the damage was much more milder. So these results led us to our second question. So our second aim was to compare the sensitivity of the central and peripheral regions of the retina after ischemic injury. In addition, we are curious about whether it is worth focusing more on the peripheral regions during the diagnostic procedure. So we reevaluated our OCT measurements again to compare the central and the peripheral regions. So we could work with 231 measurements containing data about the central and peripheral regions separately. So this is the PDF that the OCT generates. So the software analyzes the layers, so exactly the average thickness of the given layers, but we can calculate the thickness manually in the central and the peripheral regions. So this is the mid-periphery, not the far periphery, according to the basic settings of our OCT device. So all retinal layers were analyzed again, but I would like to highlight the photoreceptor layer, especially the outer segments of the photoreceptor cells, because the photoreceptors are extremely prone to ischemia. So these graphs are similar to the previously shown OCT figures, but you can see the central and the peripheral regions separately. The red line shows the control group, while you can see the ischemic groups with blue and green color. And we analyzed just the main effects because of the many variables, and we observed a significant decrease in the thickness over time, so during our experimental period, and we have a difference between the central and the peripheral region. However, the interaction was not significant. This means that we cannot say that the damage was more severe in the central or in the peripheral region. This was in contrast with our immunohistochemistry results, so photoreceptor labeling was also performed. And you can see the cones with green, the rods with red, and all nuclei with blue staining. And we observed a cone loss in the UCCAO group, which was much more pronounced in the peripheral regions of the retina. But these are just preliminary results, so we have to increase our sample size. But after quantification, this means a significant increase in the UCCAO group in the peripheral region, but not in the central region. So how can we conclude our results? The 20 minutes BCCO is a good model for investigate the retinal hypoxia in a time-dependent manner, and this is a good model for finding the most sensitive cell types. In addition, the UCCO causes more severe damage in a shorter time, so this model can be used for testing potential new drugs. And these results are already published. In addition, based on our preliminary results, the damage probably starts in the periphery and is more severe there. However, this difference has not yet been detected by imaging techniques also used in the clinical practice. So more research is needed regarding this question. And finally, I would like to thank my mentors, Professor Dóra Reglődi and Alexandra Váci, without whom I wouldn't stay here today. And I'm extremely grateful for our whole retina research group, because without them, this all would be not possible. Thank you for your attention.
Inez, it was a great pleasure to listen to you. Please stay there. And I also would like to ask Dóra Reglődi to come up to the stage. And I would like to tell also that during her university training, she could go to the US, also have an international program coming back. I think you already have 2 first author papers and just started the PhD. So Inez is really a role model with Dóra Reglődi, so I would like to ask András to bring the certificate for you.